Welcome to Advanced Data Analysis 2 with me, Erik Erhardt, Professor of Statistics at the University of New Mexico. In this video, we'll continue Chapter 3, A Taste for Model Selection in Multiple Regression, and talk about Dennis Cook's rat data. So here we are at the course website. Scroll down. Here we are in Chapter 3. Open up that PDF and the R file to follow along. So I'll jump over to the lecture notes. So Dennis Cook, you'll know from Cook's Distance. He's a professor of statistics at the University of Minnesota, and this is for me, a beautiful example about how important careful diagnostic analysis of regression models is, and how easily a person can be misled by their results if they do not perform model diagnostics. So I'm just going to read the experimental setup here. An experiment was conducted to investigate the amount of a particular drug present in the liver of a rat. So we've got 19 rats. They were randomly selected from a larger population of rats. They were weighed and then placed under light ether anesthesia and given an oral dose of the drug. Because it was felt that large livers would absorb more of a given dose than small livers, the actual dose of the animal that the animal received was approximately determined as 40 milligrams of the drug per kilogram of body weight. So heavier rats would receive more of the drug. And liver weight is known to be strongly related to body weight, as we'll see in a moment. After a fixed length of time, each rat was sacrificed, and the liver was weighed, and the percent of the drug in the liver was determined. So it's basically a short experiment, right? You give a rat a drug, you wait amount of time so that the drug circulates. Some gets processed in the liver. Then you freeze time by sacrificing the rat and see what proportion of that drug is in the liver. And the experimental hypothesis was that for the method of determining the dose, meaning based on body weight, that there shouldn't be a relationship between the percentage of the dose in the liver, which is the Y variable, the percent, and the other variables that we're measuring, body weight, liver weight, and the relative dose. So let's scroll down and take a look at the data set here. We're going to read the data off the website. And just for convenience of plotting, I'm reordering the columns here so that the fourth column in the data set becomes the first column in the data frame rat liver. So there's the structure. Remember that Y is the percentage dose in the liver. It's going to range from 0 to 4% or so. Oh, nope. It's going to be like 20 to 40%. Okay, so let me keep scrolling down, look at the plot. I'll zoom out just a little bit so we get all the axes in here. All right, so here's the y-axis. That's the response variable. And as you look horizontally among the body weight variable, the liver weight variable, and the dose variable, you can see that Y doesn't seem to have much relationship. There's almost a fairly horizontal scatter of points here. In each case, there's a lot of variability. Between some other variables like body weight and liver weight in this cell, you do see that positive relationship, right? You would think that both the body weight and liver weight would scale together, and they do. Similarly, the body weight and the dose, that's almost a perfect line, but that's because that was how dose was determined. We measured the body weight, we did that 40 grams per kilogram dose, and so we basically have a roughly straight line there. Just similarly, dose and liver weight, there's a positive relationship there. That's because we gave more of a dose to those with heavier livers. All right. Oh. All right, sorry, that paged up. Here we are again. So there's, if we look at the correlation between the variables, we can see that there's fairly low correlation between the response variable, the proportion, or the probability of, I guess it's the percentage of the dose in the liver relative to the other variables. We do see high correlation, say, between body weight and dose, but that's because that should actually be a perfect line. But we did only approximately give the dose relative to body weight, so it's very highly correlated. If you were to test whether any of these correlations with the percentage of dose in the liver was associated with these in terms of a linear relationship, the p-values are all large, so no relationship there. So let's fit the full linear model. Here is the full model as linear model with the response variable relative to the other three. And when we look at the summary, we see that body weight and dose are both significant when they're both in the model, though we suspect that individually they should not be because the correlations are very low. Okay, so there's some relationship there, though liver weight is not significant given that the other two are in the model. Let's perform backward elimination. And so here's the step procedure. We'll do backward selection. We start with the full model, and the automated procedure says liver can be dropped. So we drop liver. Here's the reduced model, and then that's where we should stop. And both are significant with a p-value of about less than 0.02. So that suggests that each of those variables is significant, conditional on the other being in the model. And that's a surprising result because the experiment was designed so that there should be no relationship. And when we look at the plots, it doesn't look like there's a relationship. So let's try to understand what's happening by performing a careful diagnostic analysis. So all this code just produces the plots below. And let me zoom back in. All right, so when we look at the residuals versus the fitted values, we don't see any outliers in the vertical direction. In terms of a fitted value, there does seem to be one value that's sort of extreme. When we look at the two residual plots, both of these look beautiful in terms of having no real structure. A normality plot looks beautiful, but in the Cook's distance, we see one observation, observation 3, which is really influential. And it ends up being influential because, so right, the vertical axis is Cook's distance. Here's observation 3 in the upper right-hand corner, and it's influential because of high leverage. So it's an outlier in the x direction, which is also a little strange because in this experiment, we controlled how the dose was given and so on. So wonder what's happening here. So the next step is to look at the added variable plots. So here is, on the left-hand side, we've got what's the relationship after controlling for dose. So the others here refers to the dose variable. So if you regress the response variable y on dose and look at the residuals, that's the y-axis here. If you regress body weight onto dose on this horizontal axis, these are the residuals, and we see that observation 3 is way up here. Look at the slope of this line, and imagine what the slope would be if you excluded observation 3. If you exclude observation 3, if you just look at this cloud of points, horizontal line, no pattern at all, no relationship. Same thing if you look at dose and y after adjusting for body weight. Here's observation 3, and if you look at this cloud of points, roughly a horizontal line there, with lots of vertical spread between the points. So it's almost exclusively because it is exclusively because of observation 3 that we're seeing this relationship. So let's understand this a little bit more by removing case 3 and seeing what the results are. So here I'm removing observation 3. So I'm removing row 3 from the rat liver data set, calling that rat liver 3. And here's the full model. Again, I'm going to do backward selection using the step function. And in this case, none of them start off as significant. And after 3 steps, the first step we remove that top one. First we remove dose. Okay, so dose is gone. Then we remove body weight. So it's just liver, and then we remove liver, and we're down to y tilde 1. Y tilde 1, the 1 here is shorthand for the intercept only. So the best way to predict the proportion of drug in the liver is by just computing the mean proportion. No other variable helps us explain it better. So let's take a look at some of the plots a little more closely. In particular, here's the plot of dose versus body weight. Body weight. And what we see is, so given a body weight, we selected a dose. So we get almost a perfect line here, right? There's a little bit of error just based on perhaps how well you can measure a dose until you get to observation 3. So what happened? Either we measured the body weight wrong initially, and so we gave too much of a dose because of that. Second is maybe we had a dose that just was accidentally too strong. Maybe we accidentally recorded the dose, right? That dose should definitely be down here. Now, one thing that a careful eye would have seen earlier, let me page back up to our initial scatter plot. Okay, I'm going to zoom out just a little bit. So here's dose as the last column, and here's body weight as the second row. And so this is the line that we're looking at. These axes are flipped. The x and y's are flipped from the plot we saw below. But here, that point, I'll just try to zoom in on that a little bit. It's this point right here that's causing the problem. That point should be over here to the left. And it's just that small difference. But that's part of the experiment. And because this is such, so much like a straight line, and because theory suggests what the relationship should be, the model's sensitive to deviations from that. All right. So observation 3 was something happened there. If this was my experiment, I would go back to my experiment lab book that I was keeping as I was conducting this experiment, looked over my notes to see what might have happened. People make mistakes all the time. Out of 19 rats, here's one mistake. That's roughly a 5% error. That's not too bad for humans. So the main point here is that experiment or observation 3 is systematically different from the rest. And in this case, I would definitely remove that observation and take the second analysis where we find no relationship as what the answer is for this experiment. Oops. All right. So I hope that helps emphasize how important diagnostic analysis is. Throughout this semester, we will always perform careful diagnostic analysis of our models when it really counts. Okay? And there may be sometimes pedagogically that I will skip over that because I'm trying to focus our attention on some other points. But in practice, there's no excuse for not looking at all the residual plots, all the stuff that we looked at here. Basically, this bank of plots, Cook's distance to see how influential points are, to see whether that is related to leverage, all these residual plots for patterns, is normality an issue, And these added variable plots are invaluable for understanding the relationships that exist in high dimensional space. This gives us a picture of that is otherwise hard to get. All right, that's the end of chapter 3.